favorable to organize the water like that. It works out better if the two lipid molecules are pushed into the same ice-like structure, into the same sphere. So that's what happens. As you add more and more lipids, they gather together in the center. They are surrounded by these cage-like structures, but there aren't so many cage-like structures if the lipids pack cheek to jowl like that. So that's what draws oils together. Water and oil don't mix. It's not that the oil molecules want to hang around together so much. They're just trying to avoid water as much as possible, and they can do that by clustering inside these structures. Okay, so that's what I just was talking about. So here's a picture that exemplifies that. These could interact on their own with this unfavorable kind of situation independently, but if they cluster together, they minimize the unfavorable aspects of the interaction. So these lipidy molecules end up clustered together. And that's why when you make vinegar and oil dressing, eventually it separates out and the oil all goes to the top because it's not super happy dispersed in the water. But if you're going to force it to disperse in the water, it's better off that way than individually. So that triggers the clustering effect. And that shows up in a couple key places in the cell. One is the cell membrane, the other is in proteins. We've talked a lot about protein folding into these special structures, the native structures, and how hydrogen bonds and induced dipole effects can help stabilize it. Although that's true, but people consider the main driving force for protein folding to be the hydrophobic effect. Some of the amino acids that are in proteins, remember there's 20 different kinds of amino acids, some of them have hydrocarbon-like side chains on them. Oily, in other words, or fat-like side chains. And these side chains don't like to interact with water. And so protein folding is a pattern of folding that brings the non-polar types into the center, away from the water on the outside. So these blue ones are side chains that are polar, the red ones are the side chains that are hydrophobic. So again, most people consider the hydrophobic effect to be the principal driving effect for the folding of the protein. In addition to all the benefits of hydrogen bond on the side. So that's a big important factor driving native structure. And in membranes, it's the same deal. Membranes are made out of molecules like this that have hydrocarbon tails.